So just remember the differences between the purines and the pyrimidines, or you can say purine nitrogen bases or simply purine bases and pyrimidine bases. So the first easiest difference is it is larger in the size and this is smaller in the size. This is a 9-membered base, nitrogen base. This is a 6-membered nitrogen base. This one carries a double ring, so it's a dicyclic one. This is a monocyclic one. Then in this case, nitrogen atoms are present at the 4 positions, that is first, third, 7th and 9th position. In this case, nitrogen is present at the 1st position and the 3rd position. And then the purine bases are only 2 types, adenine and guanine. And pyrimidine bases are of 3 types, cytosine, thymine and uracil. And one more point you can add, that both of them, that means all the purines are common to both DNA as well as RNA, but in the case of pyrimidine, cytosine is common to both DNA and RNA, but uracil is found in RNA and thymine is found in the DNA. Now let's move on to the next topic, and that is the phosphoric acid. So we are learning now the 3rd component of the nucleotide, and that is the phosphoric acid. So we all know phosphoric acid is represented chemically in this manner, and there are, you can mention it like this also, and there are 3 hydroxyl group and 1 oxygen attached to the central phosphorus. And now this oxygen has the tendency to acquire the electron or to pull the electron towards itself. As a result of this, it develops a negative charge over it and a positive charge over the phosphorus, and the hydroxyl groups are available for condensation reaction. Again, a biomolecule chapter topic, condensation reaction, that whenever there is 2 molecules joining together, and if it is possible that OH of one molecule, imagine 2 molecules have OH as side groups, so it is possible very easily that these 2 molecules, these 2 may come along with this one in the form of water molecule, and a bond is formed. So such type of reactions are called as condensation reactions with each other. This will cause the formation of bonds. So this is how it is possible for the phosphoric acid to involve into bond formation. Now remember one thing, not all of these 3 things are present together in nucleoside, because in 11th class you have learned differences between nucleoside as well as nucleotide also. So what is nucleotide now? First of all, nucleoside. Nucleoside is pentose sugar plus the nitrogen base. And remember, the bond formed between these 2 molecules is called as N-glycosidic bond. Now in your 11th class, you learned only 2 bonds name, if you remember. One was the glycosidic bond and the second one was the peptide bond. And the third one which came was the phosphoester bond. Now remember, whenever you say glycosidic bond, when we talk about normal carbohydrates, there will be bonds between COC, that is glycosidic bond. But whenever there is a participation of nitrogen in the bond formation, it is called as N-glycosidic bond. So same thing happens in the case of the nucleosides. In nucleosides, a pentose sugar, which sugar? Ribose or deoxyribose sugar. So this sugar will join with the nitrogen base. Now nitrogen base can be any one out of these 5. If it is DNA, this can form the bond, this can form the bond, and this one can form the bond, or this one. But in the case of the RNA, this one can form the bond, this one as well as this one. So this is not present in the RNA and this one is not present in the DNA. So when a nucleoside is formed, how exactly it is formed? The first position carbon, so you can see here first prime carbon I have mentioned, the first position carbon of the pentose sugar, whether it is ribose sugar or deoxyribose sugar, rule is same. First position carbon of the sugar will make a N-glycosidic bond with the first position nitrogen in the case of pyrimidine and 9th position nitrogen in the case of purine. So you have to by heart this rule that nucleoside formation involves the bond formation between the first carbon of the ribose sugar or deoxyribose sugar and the first position nitrogen or of the pyrimidine, otherwise the ninth position nitrogen of the purine. If you remember, we have already learned that first position, ninth position, ninth is not here. So in this case, always the ninth position will form a bond, and in this case, always the first position nitrogen will form the bond. So this is how a nucleoside is formed. So once a nucleoside is formed, how do you give it the names? Because nomenclature is in the NCERT of 12th class. If you go through the pages, the first pages will have these nomenclatures mentioned, and in your 11th class book you had some of the diagrams explained how do you name them. So if it is a ribonucleoside, NCERT only talks about this one in the 12th class, but I wish to make you understand this also side by side. So how do you name the nucleosides? So if it is a ribonucleoside or if it is a deoxyribonucleosides, rules are going to be slightly twisted, because this carries deoxy sugar and this one carries the normal ribose sugar. So whenever there is a ribose sugar joining with the adenine, remember the bond is always N-glycosidic bond. Whenever a ribose sugar joins with adenine, it gives rise to adenosine or adenosine. When a ribose sugar joins with guanine, it will form guanosine or guanosine. When a ribose sugar joins with cytosine, now don't get confused, osine, both will carry osine. Here this is a normal nitrogen base and these are nucleosides. So when a ribose sugar joins with C, it will give rise to cytidine, and when a ribose sugar joins with uracil, it will give rise to uridine. Now the rule, the easiest thing to remember here is the rule. Rule is but whenever a purine joins with the ribose sugar or the deoxyribose sugar, the ending of the name of the nucleoside would always end with osine, and whenever it is a pyrimidine, then it will end with iodine. So this is the rule which you have to remember so that you don't make mistakes.